Hey guys, welcome back to One Stop Biology. So in today's video, we are going to discuss Human Genome Project. Let me remind you that we have already covered most of the important topics of the second chapter of genetics, which is molecular basis of inheritance. And in this video, we are going to discuss, what exactly led to the project, to a huge project that was started off in 1990, which was, discovering the entire human genome, basically determining the entire DNA sequence of human beings. So basically, till now we have studied, or we have, kind of proved that DNA is something which has sequence of bases, and those sequence of bases determine the genetic information of an organism, of any organism, right? So if I say that the genetic makeup of an organism or an individual in the DNA lies in that, sequence of bases, it won't be wrong. It is correct, right? So, say if I have to say that, there are two humans and they differ in their physiology, or they differ in their anatomy, they differ in their phenotypic conditions. It's because their DNA sequences are different, right? If not at all places, at least there would be some places in the DNA sequence which would be different for both of them, right? So, exactly this assumption basically led to the question that one should actually, completely find out the human genome sequence. And in 1990, a very ambitious project was launched when in 1990 a very important project was launched, and that was to sequence human genome, right? So this project was basically named as Human Genome Project, HGP. Okay, this was a very big project, guys. This was a mega project, and you can imagine how. So basically, human genome, we say that they have almost 3 into 10 to the power 9 base pairs, right? And when, at that time in 1990, when the scientists, calculated the estimated cost to basically know these 3 into 10 to the power 9 base pairs exactly in what sequence do they occur, the cost was around $9 billion. 9 billion US dollars, right? Now, apart from that, this was just the cost to discover the entire sequence. Now, apart from that, if you are basically, discovering the entire sequence, you will have to store it in some form or the other, right? So, if you get it typed in a book, and if you say that one page can have 1,000 letters, one page of that book can have 1,000 letters, and say one book can again have 1,000 pages, then you would require 3,300 books to store the entire sequence of that DNA, right? And also, when basically the scientists, the team that was working on Human Genome Project, figured out that, the cost would be huge if you store the data offline. There was a different, branch of biology that was started, which was basically using high speed computational devices for data storage, right? Because if you store it in a computational device, you can not just store the data easily, but you can retrieve it anytime and you can analyze it, right? So that is where a new branch of biology was discovered, which is bioinformatics. Again, a very interesting and very important branch as per today's, scientists. If you check with the scientist community today, it is considered as one of the most important branches. of biology, and it is very high in demand in developed countries, right? So what exactly was the goal of Human Genome Project? Now, some of the important goals was one, the first goal was to identify almost 20,000 to 25,000 genes in humans or in human DNA. That was the first goal, right? Now, the second goal was to determine sequence of 3 billion base pairs that make up entire human DNA. Then, once you discover the entire 3 billion base pairs, you have to store this information, right? For further use. So, store this information in a database, right? Then, once you store it, definitely you are going to analyze it, right? So, to have improved tools for analysis, right? Analysis of this data. So to have improved tools for data analysis. Then say if you have to have important technologies, right? To use this data in different sectors, maybe in medicines, maybe in different kind of treatments. So you have to develop those technologies as well. So to develop these technologies, right? And then you'll also have to deal with several ethical, legal, and social issues that would arise because what you're doing is basically you are completely determining the DNA sequence of a person and you are storing it somewhere, right? Either in the form of, say, hard copies, which is books, or on a computational device. And tomorrow, what if that data gets leaked? Or you are basically devising a technology which can be used in a negative way as well. And not everyone would align to this mentality of determining the entire human genome sequence. So there would be certain ethical, legal, and social issues, and one will have to basically address all these issues as well, and that again would cost something, right? So these were some of the goals of the Human Genome Project. Now, Human Genome Project was basically a 13-year project, right? So it went on for 13 years and it was coordinated by one, the US Department of Energy and the National Institute of Health. So both these institutes, both these departments basically coordinated for this project. Now during the early years, the Wellcome Trust, which is there in UK, became a major partner, and then came contributions from Japan, France, Germany, China, right? And finally, this project was completed in 2003. So it started off in 1990 and it was completed in 2003, right? Now, what happened after that? So basically, after this project got completed, and we basically determined the entire sequence of human genome. This knowledge was, you know, a revolution, and it was used in many ways, right? It was used to diagnose diseases, to treat diseases, to even prevent certain disorders from happening in human beings, right? Now, apart from that, it also led to then determining sequences of other organisms as well, like say certain bacteria, viruses, yeasts. It can be a pathogen to plant, to animals, right? So all these DNA sequence, all these, animals were basically discovered, and then their DNAs were discovered, the entire sequence was discovered, and all these informations were used to develop medical treatments against different pathogens. Now, what was the method that was used in the Human Genome Project? So the method involved two different approaches, right? Let me just clear the screen. Yes. Now there were two methods that were used. One approach focused on identifying all the genes that are expressed as RNAs, because RNA are the RNA sequence is the one that determines the proteins, right? That codes for the proteins. So there were two methods. One was identifying all genes coding for RNA. RNA here was referred to as the sequence that would express itself. So expressed sequence tags, ESTs, right? Now the second approach basically was considered as a blind approach, and this would simply sequence the whole set of DNA consisting of coding plus non-coding sequence, right? So first you sequence the whole set, sequence the entire set, which is coding plus non-coding, right? And then after determining the entire sequence, you assign different regions in the sequence with respect to functions. So basically after this, you give, you annotate. So you do sequence annotation. Right. And how it was done? So basically, what first was done is, uh, first for sequencing, the total DNA from a cell was isolated, and that was converted into random fragments of relatively smaller size. Right. So if you see this diagram, right, so this is the representative diagram of HGP. So first what you do is you just isolate the entire DNA. So first is isolating DNA, and then cutting it into smaller fragments, right? Now what you do is you clone it in suitable host using vectors. Then you clone these fragments into suitable vectors, right? Now, because cloning, what you're doing basically, you're amplifying each piece of DNA fragment so that it's gets sequenced subsequently, and it would that would be done very easily because you have a suitable vector, like say you have bacterial vectors, you have yeast vectors, and they basically replicate at a very fast rate, right? So you basically clone it through, say, BACs or YACs. BAC is bacterial artificial chromosomes, and yeast is, this is yeast artificial chromosomes, right? Now these fragments were sequenced using automated DNA sequencer. Now this automated DNA sequencer worked on the principle of Frederick Sanger. So Frederick Sanger, the scientist, developed a method which basically worked on the principle of sequencing these DNA fragments. Basically, how this method worked, this used to determine amino acid sequence in proteins and then backtrack RNA and DNA, right? So so the entire sequence was sequenced using automated DNA sequencers, and then these sequences were arranged based on some of the overlapping regions present in them. So, third step is basically fragments were sequenced using automated DNA sequencer, and then sequences were arranged based on some overlapping regions, right? So you can imagine how tedious it would be, because you would be having so many sequences, small fragments of sequences, and then you would study all of them. You would find the overlapping fragments, and then you would align all these sequences together, right? So basically, it was so tedious that it was not possible humanly, to align all of these sequence. And that is why we used computer-based programs, which basically, assigned these sequences in an order, right? So the sequence of chromosome 1, the first chromosome, was completed by May 2006, right? Now this was the last chromosome that was sequenced, right? Now, again, another challenging task was basically assigning the genetic and physical map on the genome. So again, this was generated using information on, polymorphism of certain recognition sites and repetitive DNA sequences, which were known as microsatellites, right? Now, what are some of the features of human genome? So let's see what exactly was, you know, inferred to after we sequenced the entire human genome. So the first thing that we got to know was that human genome contains 3164 3164.7 million base pairs, right? Now the average gene, so a gene consists of 3000, around 3000 bases, but size of a gene varies greatly, and the largest known human gene dystrophin is dystrophin with 2.4 million bases. 2.4 million bases, right? Then another discovery was that the total number of gene is estimated at 30,000, right? And initially, when, without sequencing, scientists predicted the total number of genes. They predicted it to be 80,000 to 1,40,000, but actually it was 30,000, right? So what basically, was inferred with this information is that 99.9% of the bases, the nucleotide bases, are exactly the same in all humans, right? So again, a very important discovery after we knew the entire sequence, right? Now the functions. So basically, when after that, when they studied the entire genome, they found out that the functions of around 50% of the genes, the discovered genes, are unknown, right? And only 2% of the entire genome codes for protein. Only 2%, around 2% or even less than that, code for proteins. Now, after that, the repeated sequence. So basically, if only 2% code for proteins, what would the other 98% do, right? So there are around 50% whose function is still unknown. And then there were, a lot of repeated sequences. So there were a lot of repeated sequences which do not code for proteins. Now, these repetitive sequences are nothing but stretches of DNA sequences which are repeated many times, sometimes even 100 to 1000 times. And they have no relation with protein coding. They have no relation with coding for proteins, but then they would give, structure to the chromosome. And sometimes they would they would be very dynamic to evolution, right? So they would not code for protein, but they would give structure to chromosome, or they would be very dynamic to evolution. Now, then they basically studied about chromosomes as well. So chromosome 1, which was, the last chromosome to be discovered, has maximum number of gene, which is 2968, and chromosome Y has the fewest, which is 231 gene, right? Now, apart from this, scientists also identified 1.4 million such locations where there were single base DNA differences, right? So these are known as SNPs, single nucleotide polymorphism. These are pronounced as SNPs. So they found 1.4 million locations where there were SNPs, right? Now, this information basically makes us realize that the process of finding chromosomal locations for disease associated sequences and tracing human history has become very easy now, right? So, with this, let's discuss what are the applications and future challenges of discovering the entire human genome as well. So basically now, knowing the entire DNA sequence defined a lot of research which led to different biological systems. So it was not just about the human DNA. On the basis of that, we derived, the sequences of a lot of important organisms which were in somehow, related to humans, right? And this enabled, a new approach to biological research as well. So now, since you know the entire genome or the gene pool of humans, you can basically interconnect this with a lot of different diseases for which there was no cure before 2003, right? And that is how we started treatment of, major diseases like cancers and tumors as well, right? So, guys, with this, we have finished the entire Human Genome Project. Again, a very basic topic, but then it's very important from your boards and NEET exam point of view. So, I hope you understood the video. Please do let me know if you have any doubt. What you can do is you can either mention your doubt in the comment section of this video, or you can also send your doubts on the WhatsApp number, and I have provided that number in the description of the video. And guys, please do not forget to like the video, share it with your friends. And yes, if you're new to the channel, please do not forget to subscribe to the channel to get all the updates. So see you guys in the next video. Bye bye.